Welcome to Chapter 5, AlphaFold, the Revolution in Protein Structure Prediction. This chapter tells the story of how AI solved biology's 50-year grand challenge. AlphaFold decoded what's often called the second genetic code, how protein sequences fold into 3D structures. This breakthrough fundamentally changed structural biology and drug discovery. Let's look at the historical timeline. In 1972, Christian Anfinsen received the Nobel Prize for showing that a protein sequence determines its structure. In 1994, CASB began as a structure prediction competition. In 2018, AlphaFold won CASB 13. In 2020, AlphaFold 2 achieved historic success with GDT 92.4 at CASB 14. By 2022, over 200 million structures were released. And in 2024, AlphaFold 3 expanded to protein complexes. Consider the scale of the challenge. The human genome encodes about 20,000 different proteins. Determining just one structure experimentally has typically cost on the order of $100,000 using X-ray crystallography, and a difficult target could absorb years of work. By 2020, only about 170,000 structures had been determined, less than 1% of all proteins. This bottleneck severely limited biology and medicine. At CASB 14 in 2020, AlphaFold 2 achieved a GDT score of 92.4 out of 100. For context, the previous best scores were around 60 to 70, and experimental methods like X-ray crystallography score about 90. AlphaFold's lead of roughly 20 GDT points over the second-place group was unprecedented. CASB founder Dr. John Molt declared that a problem open for 50 years had essentially been solved. Here's how AlphaFold prediction works. You input an amino acid sequence, and AlphaFold outputs both the 3D structure and confidence scores. The PLDDT score indicates accuracy for each residue. Scores above 90 are equivalent to experimental structures. Between 70 and 90 is suitable for modeling. Below 70 indicates low confidence, often in flexible regions. The industrial impact has been transformative. Structure determination time dropped from 5 years to minutes. That's a 99.999% reduction. Costs went from $120,000 to nearly free. The 2021 Nature paper has been cited tens of thousands of times, making it one of the most cited papers of the decade. Major pharmaceutical companies, among them Pfizer, Novartis, GSK, and Roche, have publicly described using AlphaFold predictions in their discovery work. You can download any AlphaFold structure using Python. The URL pattern is the AlphaFold site at alphafold.ebi.ac.uk, then /files, then af-, the UniProt ID, -f1- model version 4, .pdb. Use the request library to download the file and BioPython's PDB parser to parse it. The function returns the structure object and prints the number of residues. Over 200 million structures are freely available. AlphaFold's architecture has 3 main components. First, multiple sequence alignment generation searches databases like BFD and UniRef for evolutionarily related sequences. Second, the evoformer uses 48 transformer-like layers with row and column attention to extract structural information. Third, the structure module predicts 3D coordinates with an equivariant transformer. The evoformer's attention mechanism differs from standard transformers like BERT or GPT. It uses pair representation to explicitly model interresidue interactions. In the MSA representation, rows are sequences and columns are residues, so row attention operates along the residue dimension within a single sequence, while column attention operates across sequences at one residue position. This dual attention, applied over 48 layers, captures both local and global structural patterns. PLDDT stands for Predicted Local Distance Difference Test. It ranges from 0 to 100 for each residue. Very high scores above 90 indicate experimental level accuracy. You can trust these regions completely. High scores between 70 and 90 are suitable for modeling and most applications. Low scores between 50 and 70 may indicate flexible loops. Below 50 is unreliable. We can extract and analyze PLDDT scores using BioPython. In AlphaFold structures, PLDDT is stored in the B-factor column. Extract the Ca atom B-factor for each residue and plot against position. Draw threshold lines at 90, 70, and 50 for reference. Calculate statistics like mean PLDDT and percentages in each category. This tells you how reliable the overall prediction is. Why is MSA so important? The key insight is coevolution. When two residues interact structurally, they often mutate together across evolution. If residue A picks up a positive charge, its contacting partner B tends to become negative in the same lineages, preserving the pairing. By analyzing patterns across thousands of related sequences, AlphaFold infers which residues are in contact. We can generate MSA using BLAST to search for homologous sequences. For each position in the alignment, calculate how conserved the amino acids are. High conservation often indicates functional or structural importance. AlphaFold actually uses much larger databases like BFD with billions of sequences, but BLAST demonstrates the concept of finding evolutionary relationships. CollabFold makes AlphaFold accessible without programming. Access the CollabFold notebook on Google Colab, enter your amino acid sequence, click Run All, and wait 10 to 30 minutes for results. It uses free Google GPUs for computation. This democratizes structure prediction. Anyone can predict protein structures without expensive hardware or deep computational expertise. We can visualize AlphaFold structures interactively using Py3DMol in Jupyter notebooks. Load the PDB file, create a viewer, and set the style. Coloring by PLDDT shows confidence, blue for high, red for low. You can rotate and zoom the structure with your mouse. This visualization helps identify reliable and unreliable regions at a glance. AlphaFold is revolutionizing drug discovery. The pipeline starts with target identification, then AlphaFold structure prediction, pocket detection using tools like fPocket, docking with AutoDock-Vina, lead optimization, ADMET prediction, and finally candidate selection. AI-driven discovery programs at companies such as Exscientia and In Silico Medicine have reported reaching clinical candidates in roughly 1 to 2.5 years, against 4 to 5 years for conventional pipelines. Structure prediction is one ingredient in those pipelines rather than the whole story. We can detect binding pockets from AlphaFold structures. Surface residues have fewer neighboring atoms within 10 angstroms. We cluster these surface residues by proximity to identify distinct pockets. The largest pocket is often the active site. While specialized tools like fPocket are more accurate, this demonstrates the concept of finding druggable sites from predicted structures. AlphaFold accelerates materials science beyond drug discovery. In enzyme engineering, we can predict industrial enzyme structures, design active site modifications, and improve stability. For biosensors, we can optimize fluorescent proteins and design binding domains. For nanomaterials, we can design protein nanoparticles and self-assembling structures. RMSD, or root mean square deviation, measures structural similarity. Smaller values mean more similar structures. RMSD less than 2 angstroms indicates nearly identical structures. Between 2 and 5 angstroms suggests the same overall fold. Above 10 angstroms means different structures. We can use BioPython's superimposer to align structures and calculate RMSD. Despite its success, AlphaFold has limitations. Flexible intrinsically disordered regions are poorly predicted. Use NMR or SAXS instead. Protein ligand complexes are inaccurate. Combine with docking or use experimental structures. Dynamic behavior isn't captured. Use molecular dynamics. Post-translational modifications aren't considered experimental validation is essential. Intrinsically disordered proteins, or IDPs, lack fixed 3D structures. About 30 to 40% of proteins contain disordered regions. They function in transcription, signaling, and molecular recognition. Examples include the N-terminus of p53 and the tau protein in Alzheimer's. AlphaFold struggles because there's no single correct structure. These regions adopt multiple conformations. AlphaFold 3, announced in 2024, added major new capabilities. It can now predict protein DNA and protein RNA complexes. It considers some post-translational modifications like phosphorylation and glycosylation. It models metal-ion coordination in active sites. On the POSE-Busters benchmark of protein-ligand complexes, AlphaFold 3 places roughly 3/4 of ligands correctly, well ahead of classical docking programs. Note that AlphaFold 2 could not handle ligands at all, so this is a new capability rather than an incremental improvement. The AlphaFold database can be searched programmatically. Query UniProt for protein names, then construct AlphaFold URLs from the UniProt IDs. This enables automated analysis of many proteins. For example, searching for p53 human returns entries for cellular tumor antigen p53 and related proteins, each with direct links to their predicted structures. We can build integrated analysis workflows with a Python class. An AlphaFold Analyzer class combines structure download, PLDDT extraction, quality assessment, and flexible region detection. Its getSummary method returns a comprehensive report. For EGFR, we might see mean PLDDT of 84, indicating very good quality suitable for modeling, with some flexible terminal regions. Looking ahead, research is moving toward dynamic structure prediction that captures multiple conformations and allosteric changes, integrating with molecular dynamics. The inverse problem, designing sequences to achieve desired structures, is advancing with tools like RF diffusion and protein MPNN. By 2030, drug development could shrink from 10 years to 3 to 5 years. Let's review the learning objectives. At the basic level, you can explain GDT 92.4 significance, the 50-year challenge, PLDDT interpretation, and MSA importance. Practically, you can download structures, analyze PLDDT, visualize 3D, and compare structures with RMSD. For applications, you understand drug discovery uses, limitations, and biomaterial applications. The exercises test your understanding at 3 levels. Easy questions cover GDT scores, PLDDT categories, and AlphaFold inputs. Medium questions ask about time and cost reduction, RMSD interpretation, and intrinsically disordered proteins. Hard questions involve target selection strategy, COVID-19 contributions, enzyme engineering workflows, and AlphaFold 3 capabilities. AlphaFold played a crucial role during COVID-19. When the SARS-CoV-2 genome was published in January 2020, DeepMind released AlphaFold predictions in early March for 6 understudied viral proteins, including ORF3A and several non-structural proteins whose structures were still unknown. The spike protein was deliberately left out because an experimental cryoelectron microscopy structure of the spike had already appeared in February. When an experimental structure of ORF3A was published a few months later, it broadly agreed with the prediction. The lesson was that structure prediction can supply usable starting models during an emergency without waiting for the experiments to finish. For next steps, try hands-on projects. Predict structures for your research proteins, analyze the AlphaFold database statistically, or detect pockets in drug targets. Learn related technologies, molecular dynamics with GROMACS, docking with AutoDock-Vina, protein design with RF diffusion. Key references include the Nature 2021 paper and the AlphaFold database. Congratulations on completing all 5 chapters of the Bioinformatics-Introduction series. You've journeyed from protein structure basics through sequence-based machine learning, molecular docking, biosensor and drug delivery design, to the AlphaFold revolution. For continued learning, consider chemoinformatics-introduction or data-driven materials design. Thank you for your dedication to learning bioinformatics.